Hi, it is my pleasure to welcome my friend and former partner in crime, Dr. Priya Banerjee, to today's Kitchen Table Talk, hosted by Dewpoint Therapeutics. Priya earned his bachelor's degree in chemistry at the University of Calcutta, and after graduating with a master's degree in chemistry in India, Priya moved to the US to pursue his PhD in the lab of Jayanti Pandey at SUNY Albany in New York. Priya studied protein liquid phase separation actually before it became mainstream cool. During his PhD, he studied mutation-induced perturbation in phase separation in crystallines and how this process contributes to development of cataracts. After his PhD, Priya moved to San Diego, where he joined Ashok Deniz's lab at the Scripps Research Institute. Here, his work focused on characterization of IDR and IDP conformational landscape using the power of single molecule fluorescence techniques. During this time, I had the honor to work with Priya, and we co-authored 3 manuscripts together. In 2017, Priya moved back to New York to join University of Buffalo, where he is currently an assistant professor in the Department of Physics. Priya's lab now applies high-resolution single molecule fluorescence techniques to study the biophysical principles that underlie regulation of RNA and protein condensates. So welcome, Priya, and we look forward to hearing about your work.
Thank you, Diana, for such a nice introduction. Can everybody see my slide? Or, yeah, great. Okay, perfect. And my laser pointer? Okay. It was great introduction. I just want to mention one quick thing. My encounter of liquid phase separation was in my PhD days. And I still remember when, as a fresh graduate student looking to join a lab, I walk into this this laboratory where they were studying CAF lenses, and they put them at 4 degrees centigrade room. The lens becomes completely opaque, and they put it outside. Within minutes, it's completely clear. I did not understand what's going on, and they told me that they're looking into the molecular details of that process. I thought it might not be a bad idea to study that process, and later that became a very important aspect of cell biology, as we now know that many cellular processes, if not all cellular processes, have some sort of phase transition driven control in space and time. So again, thanks for the invitation today. I'll be talking to you about some of the stuff that we are doing in my lab in last few years. We're looking at the biophysical principles that govern formation and regulation of such condensates. And here we're looking at molecules which are present in all these condensates, such as protein and RNA. To the audience of phase separation, I think we don't need much introduction to tell you why this is important. I just wanted to acknowledge all this work by many different laboratories over the past 5 to 7 years, showing that cells have these bodies that do not have membrane. They are now classified as biomolecular condensates. So if you go to the nucleus of a typical eukaryotic cell, you see nucleolus where ribosome biogenesis takes place, and there are chromatin domains, nuclear speckles, which are all phase separated or to some extent phase separated condensates. If you go to the cytoplasm, you have stress granules, processing bodies, which play a big role in RNA metabolism. So the overarching theme here is basically that these membreless organelles provide cells a way to activate or repress signaling processes, and they share this common theme of biogenesis, which is driven by phase separation. If you pick inside these organelles, you will see at the molecular level there are protein and nucleic acid molecules. They're in a condensed state of matter in a way that resembles a soft matter, but they're in equilibrium with a dispersed phase, which is depleted of these polymers, right? So this reversibility could be a key, because then cells could manipulate such reversibility to create or dissolve such condensates based on their need. I must admit that, when I was a graduate student, I read a lot of literature. I still read a lot of literature, although we are appreciating phase separation in biology in the last 10 years or so. There are papers that talked about phase separation as a mechanism to compartmentalize subcellular space. As early as 90s, where Harry Walter and Donald Brooks put together his nice hypothesis where they predicted phase separation is extremely common due to high crowding and confinement in cells. Some aspect of phase separation could very nicely explained by a theory put together by Fleury and Huggins in 1940s, where the free energy of this mixture, which tells you how stable this solution is. So suppose we have a polymer and a solvent. The free energy can be expressed in two terms, entropy and interaction enthalpy. Now the entropy tells you how disordered the world system is, and you can guess that when you are forming a condensate, it's more ordered than randomly distributing them. So this mixing entropy term is always unfavorable. So what drives phase separation is this interaction term, which is commonly called a chi parameter. And for a given simple polymer solvent system, the interaction parameter or chi parameter could be simplified by this mean field model, where you take into account of all possible interactions: polymer-polymer interaction, polymer-solvent interaction, and solvent-solvent interaction. And you might see that this condensate is stabilized if there is too much polymer-polymer interaction. So polymer-polymer interaction dominates this, or the polymer hates the solvent, so polymer doesn't want to spend more time with the solvent, so it goes to a condensed phase. In terms of protein-protein RNA interaction, the interactions, the description of these interactions are well known from protein chemistry, crystallography, and other literature. The protein and RNA side chains or DNA side chains could encode interactions such as electrostatics. You have a negative charge interact with positive charge. You can have pi systems that can interact via pi-pi interaction. A cation can interact with an aromatic system, which would be cation pi, and then hydrogen bonding is what drives DNA. Double helix formation could also play a role. So all these interactions are well characterized in terms of folded protein and how they stabilize their three-dimensional structure. But for disordered systems, such as protein and RNA molecules, which have these repeat sequences that do not fold into any specific type of structure, we might have to revisit how these interactions come to play to phase separation. There could be other forces, such as molecular crowding, as Danny Walter predicted in 1995. So all these together coming to play a big role in phase separation, as you might expect, and these interactions could be encoded by polymer sequences. That's why it's important to understand the driving forces so that we can characterize phase separation under physiological condition versus phase separation in a disease state. Typically, when we talk about phase separation, some aspect could be very well described by a diagram called phase diagram. So in this simple one component system, and by one component, I mean you have a polymer and the solvent, so a protein and the solvent. So this line is called the coexistence line. It separates the single phase region from two phase region. You can determine this line called tie line, which will tell you the composition of the condensates in the droplet versus in the dispersed phase. What you have on the y-axis here is the interaction strength that could be tuned by temperature, salt, or pH of the system. It could also be a function of protein-protein interaction, protein-RNA interaction that could be tuned by, say, post-translational modifications, as well as partner or small molecule binding. And again, you can get in and out of this one phase regime by just concentration change as well. So volume fraction represent concentration. Most of the systems that we study or you think about in terms of a cellular condensate, we have multi-components. So we also need to think about how phase diagrams look like. more than one component system. So we have the volume fraction you have the volume fraction of the second component here, you have volume fraction of one component here. And in certain cases, we see complexities beyond what I described in the previous slide. For example, here you see closed loop. So the closed loop suggests that within this regime 2, you have phase separated condensates, suggesting in addition to the interactions and changing all these solution conditions, you can again modulate phase separation by simple changing in concentration of the two polymers as well as their relative composition. So suppose you have protein and RNA system, you can tune their composition without doing anything else, without doing post-translational modification or temperature, salt, environmental changes. You'll still able to get in and out of this condensation, decondensation phase, okay? And that is something I'll focus on later part of my talk. And it is, you can study it experimentally by looking at this light scattering intensities, which tells you if there are condensate formation or not. And you have the ratio of the protein versus RNA here. So if you fix protein concentration, you vary RNA concentration, you could see there's condensates forming and dissolving. So this particular phenomena is known as reentrant phase transition, which could be extremely common for all cellular systems where phase separation is driven by protein-protein or protein-RNA interaction where more than one components are present. Okay, so to study reentrant transition, this work was done when I was still as a postdoc in Ashok's lab. We looked at this protein, which probably needs no introduction. It's called hydrogen atom of phase separation, right? It's a protein called fused in sarcoma, FUS. So this protein is associated with multiple different pathways of RNA metabolism. It is also mutated in ALS disease. The protein is typically a nuclear protein, but it also goes to the cytoplasm where it localizes within stress granules. The protein is an RNA binding protein. I'll spend a lot of time talking about FUS. And RNA is its natural partner. So when we looked at RNA-driven phase separation of FUS, we could see these reentrant transitions. You see this rise and fall of light scattering intensity, very simple technique, but you still get what you want to see, how phase separation is regulated by RNA. We looked at two different non-specific RNAs. These are homopolymeric poly-U RNA with very massive length versus 40 mer. We also looked at the same phenomenon with cellular RNA. We see reentrant transitions with poly-A-RNA as well. So this is basically telling you it's very promiscuous in terms of RNA binding, which existed. The evidences of RNA binding being promiscuous for FUS was already well known. We now show that they can regulate, the RNA molecules can regulate in a similar way how phase separation is regulated for FUS. And if you look at under microscope.
Can I ask a question?
Yes.
Have you looked at the influence of RNA structure? Does the RNA need to be unstructured?
So so we looked at to some extent, most of the studies are unpublished. So we looked at how RNA structure influences this process. So for example, you can look at how G4C2 type G quadruplex can regulate this. With some RNA, if you get phase separation, then you can still see reentrant. But if you form aggregated structure, then it's not very easy to dissolve them with excess RNA. And that could be a kinetically driven process as well, because for liquids, the dynamicity is very high. For solids, we could not test more than a few hours. But RNA structure not only changes how wide or how much transition window you have for these cases, you can also change the dynamic nature of these condensates.
Okay, thanks.
Yeah. Then what you have here is basically within the regime 2, you have these condensates, right? Okay. To understand the mechanism of this process, I looked at the RNA binding domain, and I'm always fascinated to see all this arginine-rich domain in proteins which bind RNA. In fact, the arginine-rich domains that we see in FUS are quite common in eukaryotic RNA binding proteins. These papers argue that RGG, RG type of motif are present in more than 50% of eukaryotic RNA binding proteins. And from the disease perspective, what I see here is this polyarginine glycine repeats and polyprotein arginine repeats, which are formed due to hexanucleotide expansion, which is C9orf72G4C2 expansion is also the same. You form these toxic polypeptides, and they're toxic because they cause the disease. They can also kill the cells if you put them in the extracellular medium. And from the perspective of all these arginine glycine repeats or arginine protein repeats, we see that they are also present in cellular proteins that are highly functional in RNA metabolism. So to understand the model of phase separation in this specific case, or to be specific, the reentran transition for this protein, we thought it is not a bad idea to look at what these arginine glycine repeats are doing. So we started with a very simple polymer, which is this third RGG box of FUS, which is 34 residue long protein. And you could recapitulate the entire reentran behavior with poly-uRNA or other cellular RNAs. And one thing we did here is we now varied the concentration of both the polymers and got a lot of turbidity diagrams like this and microscopy based evaluations of their phase separation, so that we could create something we call a thermodynamic state diagram where phase separation could be investigated in the two-dimensional concentration plane. So you have the protein on your one dimension and RNA in the other dimension. And the color intensity tells you the scattering intensity. More intensity means the propensity of phase separation is higher. And this dotted line, which gives you a look inside this closed loop, although you don't see the closed loop, you see just part of the closed loop, a half ellipse. You see how the two-phase regime, which is shown here by this Roman lated or Greek lated 2, you have one phase 1 or regime 1 here or regime 3. They're completely homogeneous mixture, but then you have this phase separated regime that's separated. And again, this arrow suggesting that you can do the entire transition from a homogeneous mixture to condensate to dissolution of condensates just by varying RNA. Okay, and we're not changing any protein structure or protein post-translational modification or salt or anything. We're just going to regulate phase separation by composition. With that in hand, we wanted to ask a very specific question. For example, we have this finite regime where you see phase separation. I'd like to know what regulates this phase separated regime. How wide can we be in this specific case, and what regulates the width of the two-phase regime? So that's one question. And the second question, would like to know that if I move around within this phase separated regime by changing concentration and composition of the mixture, does it matter? Does it change the condensate's physical properties? For the first part, we did a very simple study. So Ibrahim, a graduate student in the lab, he thought, if it is all about electrostatics, and that is because arginines are positively charged, RNA molecules are negatively charged. So many people thought electrostatics play a big role, and we thought that too. But one simple taste of that model could be replacing all these arginines with lysine, which is also positively charged. And the charges don't change if you do simple mutations, right? So in one study, Ibrahim changed all these arginines. So we took a design polypeptide where you have 10 arginines and we replaced all with lysine. We did not change the charge, and we wanted to see if it changes how wide the phase separated regime is. And we see it matters a lot. So the green lines suggest that the arginines have wider regime of phase separation than the lysine. Okay, and this is done with, again, poly-uRNA, homopolymeric RNA. And you see that when you try to dissolve it with RNA by adding RNA to these condensates, you see that you are shifting the overall boundary of dissolution almost tenfold. So there is a sequence code embedded within arginine that's missing in lysine that regulates the width of the two-phase regime. We now ask the following question: what happens if you change RNA? And the first experiment that we did is changing poly-U RNA. So poly-U RNA is a polymer of uracil, which has one membered ring. And the other class of RNA bases have two membered rings. So polyadenine will be a nice test RNA where you have this two membered ring, which provides more pi electron. And when we did that experiment, we saw even dramatic changes. So you have now arginines having this two-phase regime at some concentration, which is pretty low, 0.1 mg per ml. It becomes impossible to dissolve these condensates by adding poly-RNA, suggesting that it's not just the RNA, not just the protein structure sequence, it's the RNA sequence as well that plays a big role here. And again, lysines are wider than what you have in the previous poly-U RNA, but still way smaller compared to what arginine two-phase region look like. And in a simple turbidity plot, you could see that it's almost 100-fold or more than, I would say not 100-fold, it's like 50-fold change in the amount of RNA that you need to dissolve these condensates. Okay, so we looked at several other work that talked about how arginines could distinctly interact with RNA or DNA compared to lysine. And we also did some calculations based on quantum mechanical models. And we found that all these results point to a trend where arginines are far more sophisticated in interacting with RNA bases than lysine. And these interactions come from the fact that arginines, although it is positively charged similar to lysine, arginines have pi electrons in the side chain that can mediate cation pi and pi-pi interaction with a double membered ring way better than when you have a single membered ring, because the availability of pi system is much larger here. While you go to lysine does not have such pi electrons, which can still do some cation pi, but it's much weaker because the network of interaction here is electrostatic cation pi pi, while lysine can only do some cation pi and no pi at all. So these trends led to a hypothesis that there would be a rank order of interactions when we have these four interacting members, arginine adenine, arginine uracil versus lysine adenine and lysine uracil. And what you would see that maybe this widening of the two-phase regime is due to the short-range interactions beyond electrostatics, right? So we started off looking at how electrostatic interactions are important, and we found that electrostatic interactions may not be the only thing here. There could be short-range interactions that adds complexities to these interacting polymers, how they phase separate. So this leads us to hypothesize that arginines are much more interacting than lysine. And we must do experiments to sort of validate that as an experimentalist, that's what we want to do. So we thought it would not be a bad idea to look at condensate dynamics if we can actually see such changes. To look at condensate dynamics, we implemented a technique which is called correlative trap fusion technique. So in fusion, we're looking at micron scale dynamics. So we have optical traps where we can trap two droplets in two traps, and then we can bring one trap to the other in a very controlled fashion, and then you will see that at some intermediate length, they are gonna merge because of coalescence-driven fusion. And when that happens, trap one loses the droplet and it undergoes a relaxation. So this particular process could be detected very efficiently by looking at the forced relaxation from trap one, which offers you more than 2 orders of magnitude compared to normal video microscopy-based analysis. So that's why it's a little bit easier to do these measurements using optical trap. But at the same time, what you can do is you can measure on these droplets. You can implement a technique called fluorescence recovery after photobleaching, very commonly used in the field. So these are all, say, single protein molecules. You bleach them with very high pulse of laser. So the molecules are now irreversibly sent to a dark state. The other molecules can come and diffuse into the volume of bleach, and then you see some sort of recovery based on diffusion. Although FRAP has its own limitations, but nevertheless, it provides you some information on the nanoscale diffusion. So The bottom line is with trap induced fusion, you get some micron scale dynamics. With FRAP, you get some nanoscale motions. And what we're trying to look here is basically whether we have this arginine versus lysine differences. So in a typical experiment, this is how the thing works. You have two optical traps, you're flowing condensates, you can trap them under flow. And once you see you have a definite size that you want to work on, you can turn on their fluorescence channel, and you'll be able to now control the two traps, and you see how they're undergoing fusion. And at the same time, since all under fluorescence microscope, you could do FRAP measurements on the same droplets as well. Okay, so again, this was done to study if arginine versus lysine differences are manifested equally at a different length scale of condensates. And we see that indeed, with arginines, if you have poly-A RNA, you have relaxation time, which is 350 milliseconds, compared to lysine with poly-U RNA, which is 2.8 milliseconds. Again, they are undergoing fusion way faster than arginine-rich polypeptides, suggesting again that the interaction strengths are somehow controlling dynamics in the micron scale. We can quantify such effects and see that indeed poly-A RNA usually higher between poly and poly-U, and lysine versus arginine also are different and expected based on the phase diagram analysis I showed you. And utilizing FRAB measurements, we see that indeed for arginine with poly-A, there is almost no recovery, and we're looking at the polypeptide recovery in this case. And then with lysine, with poly-U, you see a very fast recovery, and these two are intermediate. So these particular FRAP curves follow the same rank order as we expected based on interaction. So overall, what we learned from this data that the interactions that govern phase separation also shows some trend in their in the condensate dynamics, not only in micron scale but also in nanoscale. Going from synthetic condensate to fused condensate, we see similar trend. So again, we took the RNA binding domain of fused, and with poly-A and poly-U, the diffusion, the fusion shows again 1 order of magnitude difference, and this is just the RGG3 domain of fused. And at using FRAP, we see twofold difference in diffusion rates. Now, one thing I wanted to mention in the previous slide, going back here, that we have to be careful when we analyze FRAP as a standalone, because you see there is no diffusion at all for arginine with poly-A, suggesting that it could be a gel-like condensate, which may not be true because you see they're still undergoing fusion in 350 millisecond time scale, suggesting that there are some decoupling between the micron scale dynamics and the nanoscale dynamics. So when we analyze globally FRAP fusion data, we see such trends. And we see similar behavior for fused RNA binding domain as well as the RGG3, because you see diffusion is only twofold different, while you have the fusion relaxation time almost an order of magnitude different. But overall, it tells you that the cation pi-pi-pi types of interaction further brings out regulatory elements over electrostatic interaction, and they work together to modulate phase behavior as well as the dynamics of these condensates. Okay, so with this, which was published last year, we concluded that cation pi and pi-pi interaction oppose the reentrant dissolution of protein RNA condensates, and this could be an important feature for repeat polypeptides because you add more arginine into it and you might lose the way you regulate their dissolution. Okay, so the next part of my talk, I'll be focusing on something we have done in the recent past, and it's very much a work in progress, and I'll be happy to get your feedback on this. So we talked about their phase behavior. We talked about their condensate dynamics. How about their structure? Because coming from a field of protein folding happens because there is a global minimum. And everything goes there, right? So there is one state where the structure is folded and it might go there. Disordered proteins do not fold because it's pretty flat, because there are no overall stable minimum. For phase separation in these two component systems, you see that there is a finite regime where you have condensates, okay? So it's not like you're forming condensates at a very specific composition or concentration. You are changing their composition, and you might change the properties, which we do not know yet, right? So one of the idea is to understand if we form condensates under all these different compositions, which is shown here by this dotted line, and see similar behavior. That would be pretty boring to me, but I still like to see that. So what we have done, we looked at the relevance of this problem from biology. And going beyond, just simple droplet models is necessary because you look at cellular condensates and you see hierarchy of structures. You see that, for example, in nucleolus, you have these substructures that are well described by coexisting droplet, coexisting multiphasic condensate model. Similarly, in stress granules, you see a core shell structures. Lately, we started seeing a different type of structure. For example, if you overexpress this RNA binding protein TDP43, and you find this engineered condensates in cellular in cell nucleus, which have nucleoplasm filled multiphasic structures inside. Okay, so they are called vacuolated condensates. The mechanism is not well understood, but what comes from all these analysis is the fact that condensates could have non-trivial structures, and there could be subcompartments that can be utilized further to communicate with, in the cellular environment in a way that might be important for signaling functions and control over gene expression, over stress regulation, because that's how multiphasic compartments should work. So to understand the building blocks of this process in a simplified system, we thought maybe RNA protein mixtures might play a role here. So in the first experiment that we did almost 3 years ago, we changed the RNA concentration of the existing condensates to see what happens to the condensates. And what we have found that by simple mixing experiment, if you have, so I have to play this video. Let me just escape. Yeah. So let me just play this video. So what we are doing here is within the existing condensates, we are changing the RNA concentration without dissolving them. And I hope you can see the video. So first we let them grow a little bit, and then we're going to change the RNA concentration. And what you see formation of these multiphasic structures, which are unstable because they're fusing within 5 minutes, they'll go away. But what we see that it's possible to create even transiently such structures, which you typically see in a cellular condensates. And we're again utilizing a very simplistic model system in a test tube where we have one protein and one RNA. Okay, so we spent last 2 years to chase down these structures, whether we can form them and what type of systems would form them. And we found that pretty much everything that we see undergoing phase transition by heterotypic interaction has this kind of structure. So first, what we have done here is we took another model protein system, protamine, which is this repeat arginine residues. And we created a phase diagram to see if there are specific composition of the mixture or concentration of the mixture where you see these hollow condensates. And we did that over a very broad range of concentration. So we saw that if you are too low in concentration, you don't form anything. And then if you have certain concentration and composition regime, you see these droplets, and they prevail for a really wide range. And after that, if you keep pushing the concentration, you see condensate formation, which are not your droplet-like structures. They are more like your donuts, donut-like structures. So you see that they have this ring-like appearance under a fluorescence microscope. They are much larger than what you see in a typical condensate as well, which was extraordinary to us because we are now forming these structures, just mixing protein and RNA. And when the system undergoes such a structural transition, it becomes a problem for the system because we are creating a larger surface area, because we are now creating two surfaces compared to one surface for a condensate. So we wanted to understand what's going on. But before we do that, I wanted to sort of show you a little bit more how these structures look like under a fluorescence microscope or DIC. So these are all RNA concentration higher than the protein concentration. So the protein concentration is 4 mg per ml or 4.4 mg per ml, and RNA is about 5-fold higher. And the internal lumen looks literally like a hollow condensate here. So these are Z-stack analysis of images showing that you have some sort of vesicle-like look. Here, if you start with the protein concentration fixed and keep adding RNA, your condensates size increase. And then at some concentration of RNA, they become, the polydispersity goes away. They become sort of monodispersed. And they're highly covering the overall solution, but they resist fusion. If you keep pushing RNA concentration, you see that they become very fuzzy. So they look, not very spherical. That is because they're moving around, and it's really hard to image them. And finally, even after some RNA concentration, you see these hollow structures forming. We look at under a microscope to study where the RNA and proteins are. So cyto13 looking at the RNA, and protamine is just the protein building block. And you see they both go to the rim, and the inside part, which is the lumen, sort of devoid of these structures. We can also study diffusion in specified regions, for example, in the lumen, outside, or on the rim. So here, by doing diffusion studies, we're trying to see if it is really a dilute phase within this interior space. And we see that indeed the diffusion of macromolecules outside and inside, they're in microsecond time scale in T-half, versus when you are on the rim, you see it's in millisecond time scale. So 3 orders of magnitude difference in their overall diffusion behavior in specified regions of these structures, suggesting that the internal space is indeed similar to what you have outside, which is a dilute phase. We can also do typical liquid-like property studies such as FRAP. So we're going to FRAP a small region here, and you see it recovers very well. We can also trap optically these 2 condensates and try to merge them. They form multi-compartment vesicle-like structure, and then they are merging. And then here we are trapping a droplet and trying to see if they can fuse with the rim without disturbing it, and it does actually work out pretty well, as suggesting that these phases that you see are compatible. One of the main feature of the phase diagram is to show that at specific composition this happens, right? So can we actually show that in real microscopy? So what we have done here is we trap a droplet and just change its RNA concentration. Here in this movie, you can see that 2 droplets are trapped and RNA is being flown, and you see that they undergo a structural transition to this hollow condensate. We keep them for a long time. We flow buffer. So under controlled environment where we just flow buffer, nothing happens. With RNA, they undergo this transition. We can also destroy RNA by RNase treatment. So here, over a period of 15 minutes, you see that overall this internal lumen is collapsing as RNA is being digested. And at some point, there will be a transition from condensate, from hollow condensate to spherical drop. Finally, again, is it system specific, or is it something we can see over a broad range of systems? So FUS-RGG3 does that pretty well as protamine. So this is the condensate, hollow condensate formed by FUS-RGG3. We also talked about a lot in the first part where we replaced arginine with lysine. So here we replaced arginine with lysine, and we see the condensates are still similar in structure as you have in protamine. So protamine has 21 arginine residues. We replaced all of them to create this variant called protamine K, and it still forms these condensates. So that's why we suggest that this is a property of systems that undergo phase transition via heterotrophic interactions. We also looked at synthetic condensate forming system that chemists typically study, such as this PAH polymer, which is a positively charged polymer. With poly, it forms condensates. Polyphosphate is another negatively charged polymer. Polyglutamic acid, a typical polyglutamic acid track, is found in many proteins such as NPM1, which Diana studied a lot. It also formed these condensates. One of the favorite image that I have is this with cellular RNA. So I initially thought it's not possible because cellular RNA has everything. So maybe there will be something else going on, but we can still form these structures with cellular RNA mixtures and other type of polymers as well. So overall, what we see here is a general behavior where systems under these disproportionate conditions, okay, that would be the important part for the next discussion in here, that the disproportionate mixtures drive the formation of these hollow structures. So that brings out this important question: what is the mechanism? Why some systems under certain conditions forming these structures, right? We had no clue until we found that Shklovsky published this theoretical paper in 2005, where he talked about how disproportionate mixtures can undergo condensation and form anisotropic building blocks. Now, the data I was trying to show you on systems, they are pretty much homopolymeric without any defined anisotropic structures. They are different than lipids, which are very well known to form hollow structures such as vesicles or lipid mimic polymers. What you typically do is you create a head and tail. What that means that you have a diblock nature where interactions are different among different building blocks. What Shklovsky suggested, you can still do it if you have homopolymers, but you have to drive their condensation to a partial point. So suppose you have a protein and RNA and you have disproportionate amount, you can form tadpole-like structures where part of the RNA chain, for example, is only covered by the peptide or the protein because there is not enough. So once you form that anisotropic structures, they can undergo formation of these condensates, which we call micelles. We call them micelles because now if you think about how these condensates will collapse, they can undergo condensation where these heads are joined, but their free RNA tail could be on the surface. And that might create some sort of fuzziness in structures because these condensates would not undergo fusion because of overall negative charges on the surface or repulsion. So that will lower the surface energy tremendously, so that will resist fusion. And again, it's a surface-driven property, not the property of viscosity at all. And finally, by some mechanism, these micelles can come together to form vesicle-like structures, where now these chains, which are not condensed, which are not satisfied at all, can maximize their interaction with solvent by distributing themselves over two surfaces, because you now have internal surface and you have now external surface. So what Shklovsky did here, he presented a very nice statistical mechanics model to calculate free energies and gave us some clue how to think about the process. To understand it further, if this is really what's happening, we teamed up with David and Murali at Iowa State to do some molecular dynamics simulation. So they implemented this system of rhodamine and a poly-U RNA with 100 monomeric uracil residues. And they integrated interactions which are electrostatic and Ilya excluded volume, which you typically can expect in polymers like protein and RNA chains. And indeed, they could form tadpoles at very low chain concentration. So what you see here is partially condensed head of an RNA chain and the free RNA tail, which looks like what Shklovsky presented in 2005. If you increase the total chain concentration, now you form condensates where the surface of these condensates are covered with this RNA chains, which is not satisfied. And that would explain why these condensates may not undergo fusion at all, because the overall RNA concentration on the surface will lower their overall ability to fuse because the surface energy is lowered. And finally, if you keep pushing these condensates, the RNA chains, which are not part of these condensates, will seek for more surface area, and that will create an additional surface, giving rise to this vesicle-like hollow condensate structures. Okay, so overall, the model of tadpoles work out nicely, and it also points out that the intrinsic anisotropy that you have in a lipid, it's not a prerequisite to form hollow structures like lipid vesicles. You can still form such structures by driving the condensation to a partial regime where you create an extrinsic building block such as these tadpoles in our system. Okay, so I talked about lipid vesicles. Now I think I would like to spend some time to convince you that these are lipid vesicle-like structures, although there's no lipid. One of the most important part in a lipid vesicle is that the ordering in the lipid bilayer comes from how heads are joined and tails are outside, or vice versa, depending on what type of lipid you have. And that creates the organization within this bilayer creates an ordering that can be termed as liquid crystalline ordering. So we looked for such ordering in the first place because the simulation suggests that indeed protein RNA can create some structures which could be driven by ordering in a specific way. Polarization microscopy detects such ordering. So you use polarized light, and your detectors also can detect polarized light. And if there is specific polarization that comes from refractive index mismatch, you would be able to see some patterns which are analyzed based on some models. So for liquid crystalline materials, which are hollow vesicles, you typically see this Maltese quadruped structure, which tells you that there are ordering on the rim. Under a fluorescent microscope, it looks like hollow, but if you turn on the polarization, you see this beautiful quadruped structure suggesting there are ordering of ordering of protein and RNA molecules within the rim, similar to lipid-like molecules. And you can see that also for excess protamine case. So we can not only form vesicles with one building block, which is RNA being in excess, we can form the same type of vesicles or similar vesicles where we have excess protein, lower amount of RNA, and we see similar type of behavior in polarization microscopy. Okay. And again, we do not really understand this behavior at the molecular level yet. So we are doing more experiments and simulation to understand this. But this is remarkable given that none of the building block has any sort of ordering. But overall, they can come together and create an ordered assembly. Another very important property of a lipid vesicle is a size-dependent filter-like property, right? So what you have here is basically this vesicle-like rim. When you have for lipid or any other building block, it can only let some molecules to go through while others are not permitted based on their sizes. So we thought maybe we should test that and see if there is a typical mesh size of this condensate ring. And what we have found here is using dextran probes. So dextran is a polymer that can be found in different sizes. So we went all the way from 4,400 mar to 155,000 mar. So these are different chain lengths and molecular weights. And they have typical sizes. So for example, a 10K polymer would be 2.5 nanometer in terms of their overall size. So when we look at their partitioning, when we add these fluorescent molecules on outside and see if they can go through this barrier to inside, we see that for 4.4K, the intensity outside versus inside are pretty similar. And these are again summarized right here. So the red is the lumen intensity compared to what you have outside, and the blue is on the rim. And the rim is glowing a little bit more compared to what you have, but nevertheless, it goes through. For 10K, we see that the inside is a little bit less bright compared to the outside, but the rim still gets something, and 10K falls right here. But above 10K, we don't see anything going on the rim, and you could see that 40K, 70K, and 150K pretty much similar in range. So they don't even go to the rim, suggesting that there is indeed a size-dependent filter behavior here. And the typical size based on this experiment we find is 2.5 to 2.8 nanometer. And finally, for any lipid-like vesicles, you were to see if we can encapsulate biomolecules to carry out a specific function, if you're thinking about biology or for people who are in bioengineering, they would like to deliver things. So you have to encapsulate something, a cargo maybe for that purpose. So we tested the ability of our vesicle-like structures to do the same. And we see that indeed it can do something similar. So if you have a single-stranded DNA, and this is basically the probe molecule or the cargo molecule, it goes inside the protamine like vesicles, and you see these beautiful core shell-like structures. Double-stranded DNA also goes in, and these are the profiles. You could clearly see how they look like, green versus red. Molecules such as BSA goes inside as well, and you could see that it's excluded from the rim, but included in the lumen. Also, you have prime domain of FUS. It goes barely inside, as you could see from here. That partition is not very strong. And a control substrate such as green fluorescent protein doesn't go in at all. It always stays outside. And here the inclusion coefficients are sort of ranked. So double-stranded DNA is most in our case, and then followed by the PSA and then single-stranded DNA. Again, we do not know the molecular rules that govern such partitioning yet, but we're just trying to see if our vesicle-like enclosures indeed behave similar to what you have in lipid vesicles. And we see a lot of similarities here in terms of encapsulation of biomolecules. Okay, so these data are available at our in bioRxiv article, which was also submitted in the journal for publication. With that, I'd like to summarize the part 2 of my talk, that we can create vesicle-like enclosures by phase separation, and these are completely lipid-free. And these enclosures behave similar to what you see in lipid vesicles, such as local ordering, size-dependent permeability, and selective encapsulation. Now, one of the major questions we need to understand to go forward is how cells or how biology in general can utilize structures to carry out some function, and that's something we will be we're doing right now to understand how multiphasic condensates in general can facilitate signaling reactions in general, or any biochemical reaction in general. With that, we'd like to acknowledge people who were very instrumental to do all this work that I mentioned today. So 2 postdocs in the lab, Mahadi and Richu, did the bulk of work. Ibrahim and Taran did a lot of work, which I talked to you about today, and our collaborators, UB and outside UB, or funding agencies at NIH and NTUB. And thank you again, all for giving me the opportunity to come here through Zoom and give the presentation. I love Condensate website, as you mentioned, and it's like one of the most educational thing I have ever seen in this field. So thanks for doing it. And again, my undergraduate students such as Andrew and Hannah, they also had very good impression of this website. So again, thanks for your attention, and I'd be happy to answer any question that you may have.
Thanks, wonderful.
Thank you.
I have a question.
Yeah.
Hi, this is very nice, thank you. I had a question about the RNA concentration, where you compared like different type of RNA, and you also have like cellular RNA where you see the reconstruction of the hollow condensate.
Right here,
Do you see differences when you put cellular RNA? Is that concentration lower or higher?
Yes, so cellular RNA forms this structure at a lower concentration. We also see a lot of differences in the size and the size of the rim compared to what RNA we use. So the cellular RNA that we used here is only, I think, 6 to 8 mg per ml, and that was good enough to form these structures versus, when you use poly-RNA, it was like more than at least 11 to 11.5 mg per ml. So depending on what RNA you use, yes, absolutely, you see a lot of changes.
And is the structure, the size of the condensate also kind of smaller with cellular RNA?
Yes, that is true. So with cellular RNA, it is smaller than average size is smaller than what we see for the poly-U or poly type of RNA.
Great, thank you.
Yeah.
Hi, Priya.
Hello.
Very cool talk. I love the videos.
Thank you.
I have a question about the partitioning of the different dextrans and also the specific like BSA versus DNA, et cetera.
Yes, yes.
I'm wondering how the, so these like size cutoffs, how do those correlate to the specific partitioning of the different like BSA and single-stranded DNA, et cetera? Do the other ones break the rules here because they have features, or yeah?
Yeah, so for the dextran probes, we sort of, think about these probes not interacting with the system at all, right? So because, they are supposed to be inert. So the experiments that we have done here is a little bit different, here versus the encapsulation. So here we form the condensate and we add the dextran outside and ask the dextran, can you go inside? Right, and that would be the true size dependent partitioning behavior, right? And you see that for 4.4K, 10K, it can go a little bit, but for higher order, it does not. For encapsulation, what we did, we had our cargo molecules in this in solution, right? And we formed the vesicle, and then we sort of flush out the system to see if we can still retain them, right? And typically that's what you do. We haven't done like, complete separation of the outside medium, but we see that when we do that, we can still keep some of these molecules trapped within these. So typically, to answer your question, these two experiments were done in two different ways.
Right. So if you had to guess, if you were to add these different things after the droplet was already formed, do you think it would look similar or different?
That's a great question. One thing we see is very different. So you see a correlation between what goes on the ring versus what goes inside. So for example, in dextran 4.4K, you see a spike. That's because it goes inside as well as it goes on the rim. And you can actually see that the blue and the red have some sort of correlation. It's higher, lower, and so on, right? So if it doesn't go to the rim, it doesn't go inside. That's the bottom line. Here, we see something very interesting. So for example, if you take BSA, you could see that the rim is completely dark compared to outside or inside, right? So the rim, there is a dip. So I don't think if you add BSA outside, it's going to go inside because it doesn't go through at all. It doesn't go on the rim. And that might be why it's being trapped here, because it just cannot go through it. And that could be due to interaction or due to just exclusion, that we do not know. And we think the way this is behaving is different than the dextran.
Awesome, thanks.
Yeah.
I'm really curious if you think that these vesicle-like structures may have some physiological role of trafficking within the cell.
Well, yeah, I can only speculate without data, and I would love to. But so what I would suggest is there are not much literature about these structures being forming. But in germ granules, I think there are a couple of papers on Oscar condensates where they see that the Oscar protein, which binds to RNA, can form the hollow condensates, which they call germ granule, right? And they might, they play a big role in trafficking of mRNA. And one of the defining, feature of that particular work to us was when they mutate the protein to, and reduce the RNA binding affinity, those structures don't form. Okay. So we think that there might be a lot of roles in how storage and RNA metabolism could be regulated by forming structure, because it offers a different microenvironment compared to what a condensate offers because of the high density of macromolecules within a condensate versus the lumens are basically a dilute phase inside. And yeah, so I think they might play a big role. Again, without much data here, I may not be able to go anywhere beyond speculation.
Thanks, Priya. That was excellent.
Thank you.
I was curious about. I think it's really fascinating. You were talking about the localization of the tails that are from the tadpoles. I'm just curious. if you can comment a little bit about the
Yeah.
preference, why would the tails prefer to go inside versus outside, and where a switch might happen? Like, for instance, you were showing that if you tune the ratio between the RNA and the protein, you go through hollow condensates, full condensates, and back to hollow. Do you know if during that transition, there's also a change in the directionality of the tails now that the tails are actually RNA or protein?
Yeah, so that's a great question. We probably do not have data again or simulation to answer your question, but I'll start off by saying when we looked at these structures and talked to some of the colleagues who are working on lipid vesicles, lipid rafts for a long time, they didn't believe that these are, they thought, it's an April Fool joke. These are forming by lipids. And that made us to think about and go into the literature of lipid mimicking polymers that form vesicles. And there are protein vesicles that are formed mimicking lipid-like structures. And what they have done typically is creating this, two half of a polymer, right? And the two halves, they call it diblock polymers. Two halves have different interaction propensity or different interaction with the solvent. One way to think about, Rohit Pappu published a lot of work on how differential solvation could drive phase separation versus gelation. We think that is a fascinating idea to be explored here, because if you think about this tadpole, so I'm hoping you can still see the slides, right?
Yes.
Yeah. So if you think about that, you only have condensed the head partially. So this is electrically neutral condensate. So the interaction with solvent would be different than this charged tail. And if you have to think about it, you might think that the charged tail will interact solvent more favorably than this specific part where you have a protein RNA complex. So when they are joined together, you might see a preferential orientation of these molecules or these complexes, right? Partially condensed complexes, to maximize how they interact with solvents. And that might actually tell you how they should orient. For example, when they go to the surface, the RNA chains can maximize their interaction with the solvent. And when you have higher concentration of these chains, they now can even have another surface where the RNA chains coincide. And we actually see both in our experiment and in simulation, again, that's not part of this manuscript that we presented, but we see that RNA a little bit higher in concentration compared to outside to inside, and that is because of this orientation due to favorable interaction with each other or with the solvent. But how this transition occurring from one step to another, we do not have a clue, to be honest here, because this is a fascinating phenomenon that we just found, and we're trying to study everything that we can to understand the mechanism.
So earlier you were showing that at least the diffusion is the same in the lumen and the outside. So do you expect that the composition, I don't know, salt concentration or whatnot is different in the lumen, or is more of
the
a
RNA concentration, right. So RNA concentration, so we do not know if the salt concentration is different. The FCS cannot detect the differences to be more specific, but when you look at the intensity profiles, we see a little bump inside than outside, and the simulation also showed the same thing. And that is something that we're studying right now to understand if it is indeed more enriched in RNA inside than outside. And if that is true, then it might be a way to pack RNA inside. So, that's something diffusion cannot detect. But when they did some simulation on mean square displacement of a probe particle, they put, that's also part of the manuscript. So what they found that they can detect, since it's simulation, they can detect that the outside is more mobile than the lumen. And combining with the microscopy measurement that we had, we see that the outside, although the intensity is pretty low, compared to the rim, the outside is versus inside, there's a ratio of 5:1. But again, the intensity inside is pretty, pretty low, because again, they look hollow, and it's basically you're counting darkness. But you can Also see right here, there's a little bit more red than what you have outside. The signal has to be quantified. So we're trying to do some state analysis where you can quantify the signal from inside versus outside. But the lumen actually is a problem because it is so bright, and you need a larger structure to look at those.
That's really interesting. Thank you.
Yeah.
The other thing that's interesting when you think about arginine versus lysine, you also think about different post-translational modification
Yes.
opportunities as well. And so it's always interesting, when thinking about results like yours, to then add on the layer of, well, what if some of the arginines are methylated versus not?
Right. Right.
That sort of thing.
Yeah. So we were starting with a colleague here who specializes on arginine methylation with several arginines in yeast and in human, and we see some results that has striking similarity with our arginine versus lysine. So when I was describing this data to him, he thought it was really interesting, and we looked at some of the stuff that he's doing, and we see a nice correlation. And again, unpublished data, so I can't talk a lot, but we think arginine versus lysine could be relevant to understand arginine methylation for arginines. It may not alter the phase separation by a great deal, but it can change how condensate dynamics is regulated.
Yeah, the strength of the interactions, if you have an arginine that's mono or dimethylated, the pi-pi stacking interactions can be quite different.
Yes.
In the mono and the dimethylated arginine versus the normal arginine.
Right, and at the same time, you may not change the electrostatics. So basically, you are changing arginine to say lysine, where you are selectively eliminating only one or two in the hierarchy without effectively eliminating phase separation.
Exactly, and it can slightly change the dynamic motions. It can change the strength of the interactions.
Yeah.
So the dynamics of the complexes that are forming and unforming, all of that can be affected just by a few methyl groups.
Yeah, yeah. So and that gives you a handle to regulate functions.
Exactly.
Well, excellent. Does anyone else have any questions for Priya?
Sometimes we think of questions later, so maybe we'll get back to you later.
Definitely, no problem. I'll try to do my best to email back as soon as possible. But I think there is a chat in the chat window. I saw something.
Oh, someone who had to, yeah, someone's thanking you for your talk.
Oh, okay. All right. Perfect. Thank you.
Matthias from our Dresden lab.
Oh, okay. Perfect.
Yeah.
Yeah, thank you very much. This was excellent. And yeah, we'll get back to you with more questions.
Thank you, Deanna. Thanks for the opportunity again, and a very nice meeting with all of you. So stay safe and healthy, and hope this will be over soon.
Yeah, likewise. Thanks so much.
Yeah.
All right.
All right. Take care.
Bye. It was good seeing you. Bye.
Thank you, Priya. Bye-bye.
Okay. Bye-bye.